Hi everyone, I'm Vikas Kumar. When we look at the data on deaths caused by diseases, cancer stands out as the second leading cause of death, right after cardiovascular diseases. It's responsible for around 10 million deaths each year. Among the different types of cancer, lung cancer is particularly deadly, causing about 2 million deaths. This high mortality rate is largely due to how quickly lung cancer progresses, how fast it spreads, its high level of invasiveness. One of the key factors in cancer progression is a process known as angiogenesis. So what is angiogenesis? Simply put, it's the process of forming new vessels from ones that already exist. For cancer to grow beyond a tiny size, about 2 millimeters, it enters a state where it lacks oxygen, known as hypoxia. To overcome this, the cancer releases chemicals, mainly something called VEGF, or vascular endothelial growth factor. This VEGF signals the body to start forming new blood vessels that supply the tumor with the nutrients it needs to grow. Researchers have been trying to slow down this process by developing drugs that target VEGF. Our research problem and objectives. This brings us to our research problem. We're working on developing a multi-scale hybrid model that integrates angiogenesis into tumor growth. The goal is to study how cytotoxic-T cells, which are part of the immune system, interact with cancer cells when anti-angiogenic drugs are used. During this internship, we have a few key objectives. First, we want to simulate how tumors grow and how immune cells interact with angiogenesis. Next, we aim to predict how tumors might respond to the drug sunitinib at different levels of effectiveness. Finally, we plan to analyze the behavior and dynamics of tumor and endothelial cells with and without drug treatment. The initial model. Now let's talk about the model we're using. It's a hybrid model that combines different types of modeling, continuum and discrete, at two scales, the tissue scale and the cell scale. On the tissue scale, we're looking at how nutrients and VEGF diffuse through the tissue. At the cell scale, the main agents are the tumor cells and the endothelial cells, which line the blood vessels. We've also integrated immune cells at the cell scale and drugs at the tissue scale. The drug we're using inhibits the endothelial cells, preventing them from forming new blood vessels. Meanwhile, the cytotoxic T cells target and kill the tumor cells, simulating what happens during immune therapy. Model rules and assumptions. There are several rules and assumptions in our model. First, we've defined how cells switch between different phenotypes or states. Quiescent cells or resting cells can start to proliferate or divide under certain conditions. Once they do, they divide after a specific period, and the new cells grow until they return to a quiescent state. Quiescent cells can also undergo apoptosis, which is programmed cell death, or they can enter a hypoxic state if they don't get enough nutrients. In a hypoxic state, cells secrete VEGF to try to induce angiogenesis. But if the nutrient levels improve, they return to a quiescent state. If things get worse, they become necrotic and die. We've also made some assumptions about how immune cells behave. The number of immune cells increases with the number of tumor cells. These immune cells move toward VEGF gradients, just like endothelial cells do, and when they encounter tumor cells, they kill them with a certain probability. And then there are rules for endothelial cells, which form blood vessels. They can exist in three states: tip cells, stalk cells, and phalanx cells. The transitions between these states depend on the local concentration of VEGF and the distance between cells. Stalk cells are the ones that actually divide and help the blood vessels grow. Finally, we've incorporated drug inhibition into the model. The drug, sunitinib, prevents phalanx cells from switching to tip cells, which slows down the angiogenesis process. It also inhibits the proliferation of stalk cells, making it harder for blood vessels to grow. By increasing the VEGF threshold, we've reduced the switch from phalanx cells to tip cells, which in turn hinders vessel formation. Now let's discuss the results. Initially, we ran some simulations using the basic model without any added complexity. We used the Limbesh package in C++ for these simulations. The first simulation showed that as soon as blood vessels reached the tumor quickly grew in size. But when we introduced drug inhibition into the system, we saw that the formation of blood vessels was hindered, the tumor size decreased, and there were changes in the nutrient and VEGF gradients. When we combined drug therapy with immune cells, the results were even more promising. We saw a significant decrease in tumor size because the immune cells were effectively killing the tumor cells. This combination therapy was much more effective than using either treatment on its own, as it greatly impeded angiogenesis and reduced tumor size. Population dynamics. Initially, as cancer enters a hypoxic condition, the number of hypoxic cells increases suddenly because they consume all the nutrients provided. As the initial nutrients are depleted, the cell number starts decreasing. However, once the angiogenesis process begins and cells receive a full blood supply and nutrients, there is a sudden drop in hypoxic cells. As tumor proliferative cells divide rapidly and the cancer spreads, the core of the tumor becomes hypoxic again due to the sudden increase in tumor size. More blood vessels form, leading to another drop in hypoxic cells. When comparing the original model to the therapy and drug treatments, we observe a delay in angiogenesis formation, shifting the peak of hypoxic cells to the right. The extent of angiogenesis is also reduced, resulting in a smaller drop in hypoxic cells. The most significant delay is seen with the combination of immune integration and drug therapy. By keeping the cells in a hypoxic condition for a longer time, more cells die by switching to the necrosis state. Additionally, the stalk cell count, which is responsible for the length of blood vessels, has not changed much in the case of immune integration combined with drug therapy. This indicates that angiogenesis is greatly hindered under this combination therapy. Early on, there wasn't much change in the total cell population, but after around 20,500 time steps, drug therapy, particularly the one altering stalk cell proliferation and VEGF threshold, showed better results than immune therapy alone. However, the most interesting finding was that when drug therapy was combined with immune therapy, the most significant reduction in total cell count, particularly in tumor cells, was observed. As drug concentration increases, the efficacy also increases, leading to a more significant reduction of endothelial cells, which are part of the vessels involved in the angiogenesis process. This reduction in endothelial cells directly affects the tumor's ability to grow and spread, because the tumor relies heavily on these newly formed blood vessels for oxygen and nutrients. The higher the drug concentration, the more effectively it inhibits angiogenesis, thereby starving the tumor and limiting its growth. Compared to using either treatment alone, the combination of drug therapy and immune integration proves to be far more effective. This synergy leads to a substantial reduction in tumor size and overall cell population, including both tumor and endothelial cells. By delaying angiogenesis and keeping the tumor cells in a hypoxic state for longer periods, the combination therapy increases the likelihood of tumor cell death through necrosis. Moreover, by fitting the total cell count data from each treatment scenario to an exponential curve and applying the Lowy additivity model, we can mathematically demonstrate this synergy. The analysis shows that the total live cell population is significantly lower when drug and immune therapies are combined compared to when they are administered independently. This further supports the conclusion that the combination of these therapies is the most effective strategy for inhibiting tumor growth and angiogenesis. So, in conclusion, our study demonstrates that combining drug and immune therapy has a significant inhibitory effect on both tumor angiogenesis and growth. This combination therapy not only delays angiogenesis, but also keeps the tumor cells in a hypoxic condition for a longer period, leading to more cell death through necrosis. As for the future, we plan to refine the model further by including the migration of cytotoxic-T cells, defining drug concentration gradients more precisely, and conducting parameter sensitivity analyses. Thank you for your attention. Contribution to the project was to analyze and set new rules for the agents within the model. I thoroughly understood the process and generated data through simulations, which led to the plots we've discussed. I also analyzed migration, cell-to-cell interaction, phenotypic switches, and proliferation, conducted a literature review, and identified synergy observations through data fitting.